righty. Let me introduce myself. My name is Randy Haddock. I'm on staff with the Cahaba River Society. The Cahaba River Society is a nonprofit citizen advocacy group whose mission is to restore and protect the Cahaba River and the rich diversity of life that relies on it. Now, that diversity includes the people who drink water from the Cahaba through the Birmingham Waterworks Board's system, as well as the well-known rich diversity of aquatic life that lives there. Now, the Cahaba River is located in central Alabama, a state that's already noted itself for its remarkable aquatic biodiversity. We have one of the greatest numbers of native fishes, mussels, reptiles, crayfish, and snails of any state in the U.S. Now, if you'd like to gain a deeper understanding of exactly why Alabama supports such an outstanding diversity of life, I'd urge you to find a copy of Dr. Scott Duncan's excellent book called Southern Wonder and Alabama's Surprising Biodiversity. But even among rivers in Alabama, the Cahaba stands out as an outstanding example of its rich aquatic wildlife. It probably retains a greater proportion of its original complement of aquatic wildlife of any Alabama river. We have 128 different native fishes, 36 mussels, 17 crayfish, and over 30 snails, and quite a number of reptiles. This gives you a good idea of the relative species richness of the Cahaba River compared to a couple of other much larger river basins out west. The Colorado River watershed and the Columbia River watershed are both about 250,000 square miles, which is, of course, far more than the 22,000 square miles of the Cahaba River watershed. Now, the Cahaba, as I mentioned before, has 128 fish species, including a lot of colorful darters and shiners, and of course the odd little paddlefish that we see here, the blue sucker, the blue nose shiner. That we'll go over these next, some of the fishes of the Cahaba. Dr. Rick Maiden and Dr. Bernie Caheda assert that the Cahaba probably has more fish species per mile than any other North American stream of its size or crater. So here are some examples. This is a long-ear sunfish. It's quite a showy guy. This is the iridescent blue that shows up in this fish is pretty striking in that deep orange background. That's a fish that's in the Cahaba, but it's in a lot of other streams in Alabama as well. This, I'd have to say, is an inadequate photo of the rainbow shiner from the Cahaba in Trussville. In the sunlight, and especially during breeding season, these fish are simply brilliantly iridescent. In fact, here's another photo of somebody who managed to capture that iridescence of the rainbow shiner. Now, most of the fish that I've described so far have been small darters and shiners and things like that, and they are especially colorful in the late winter and early spring months, like right about now, as we do this in early April. But this larger fish is a river red horse, and it has a pretty brightly colored red tail, and tail fin, dorsal fin, and anal fin. So it's a pretty showy fish as well. Here we have Jim Brown and Dick Mills. Jim Brown is a retired history professor from Stanford, and Dick Mills is an international ecotourism guide. They're on the Little Cahaba here, and they're using these long poles, and from the end of that pole is a noose or a lasso. And what they're trying to do is, as the river red horse are on these gravelly shoals trying to spawn, they're trying to guide this noose down and slip it over the head of the noose and capture it that way. This is a Native American technique that's been used for many generations, and there are a couple of families in Big County that keep this tradition alive. So if you are successful, it's not an easy thing to do. I've tried it and I've done it. This is my success in capturing a red horse using the noosing method. As you can see, the wire tightens behind the gills and in front of the pectoral fins, and it doesn't hurt the fish. So it doesn't really hurt the fish, and you can release them unharmed altogether. Now, here we have a photo of the record holder for a fish caught in fresh water. Now, this is not strictly a freshwater fish. This is a Gulf sturgeon, a 360-pound sturgeon that was caught in Centerville in the Cahaba River at about 1941. Now, this is a fish that generally lives in the Gulf of Mexico, but there are a number of marine fishes called anadromous fishes that like to run upstream into fresh water to lay eggs. And the Gulf sturgeon is one of those that prefers laying its eggs, has to lay its eggs in freshwater systems. So most of the anadromous fish runs that we have been lost because of impoundments on the rivers, particularly the bigger rivers. Those impoundments don't let the fish run far enough upstream so that they have limited the spawning habitats available to them. And I want to mention here another biological phenomena that has been lost due to impoundments of the bigger Alabama rivers. And that's the mullet runs that historically occurred as far upstream as Centerville on the Cahaba River. These runs were probably feeding runs, not spawning runs, but the town of Centerville used to have a big celebration where everyone in town got to go down and chow down on the fresh supply of mullet. Another of Alabama's and the Cahaba's remarkably rich group of critters are the freshwater mussels. We have 182 different species of mussels in Alabama, and they are a pretty interesting group. This is a map which shows the number of freshwater mussel species by state, and you see Alabama there is a clear leader in having 182 freshwater mussel species. Now, freshwater mussels are related to the marine clams, and like their marine cousins, freshwater mussels survive by drawing water in through a siphon. They draw that water in, it flows over their gills, and then the food particles are captured on that gill surface, and then the water is expelled through an X-current siphon. So they have an in-current siphon where water goes in and an X-current siphon where the water goes out. And so they spend you find a mussel on the riverbed and he's just kind of laying there and drawing in water and expelling that water. It's not very exciting, but it turns out that these guys have a really interesting life cycle. And that's what we'd like to describe here. Now, living in rivers presents a kind of a special problem. Rivers have this sort of habit, if you will, of always flowing downstream in one direction. And this is a real problem if you're going to expel your larvae out into the water column. These would wash downstream, and if they were to survive, then the next generation would wash further downstream, and so on and so forth, until the whole population of mussels had been simply washed away downstream out of the system. So mussels, just like every other river species, need a mechanism to colonize upstream habitats. Now, this process is an easy thing for fish to do. They simply swim upstream and repopulate those upstream habitats. It's an easy thing for aquatic insects to do because the vast majority of aquatic larvae have adults that can fly, and they disproportionately fly upstream before they lay eggs, so they repopulate the upstream parts of the stream. But mussels are not very adept, an adult mussel are not very adept at traveling upstream. They have a foot and they can move, but they really can't cross habitat that they're not adapted to. So they have to find some other way to do that. And the general solution for how they do this is to parasitize a fish and get that fish to carry their babies upstream for them. So in this diagram, we see how male mussels, here at D, this is a male mussel, he's expelled sperm into the water column, and if a female mussel draws up that sperm through the incurrent siphon, she can use that to fertilize her eggs and grow and nurture uh, her larvae in a marsupium, and then release those larvae called glochidia into the water column. And you got to release quite a large number for this to work, because these must simply randomly bump into a fish and quickly latch onto the fish, where the glochidia then transforms into a juvenile mussel. And after 3 or 4 weeks or so, once the juvenile is fully formed, it breaks loose and falls down and falls back on the riverbed. And hopefully by this time the fish has moved around enough or has moved upstream enough to keep the population of the mussel engaged in or still existing in this river. So that's a pretty clever thing to do. And this is the fellow who first recognized this life history phenomena of the freshwater mussels. This, you might have recognized this guy. This is Anton von Leeuwenhoek. He's the dude that made the first successful microscope that you see here on the right. He made drapes, but he needed a way to examine the threads of the draperies more carefully. And so he invented this microscope, and then turned his attention to all the wonderful things that he was finding in water and in other places that lent themselves to investigation for the very first time under a microscope. Now, he made this connection between glochidia and parasites of the fish and freshwater mussels. But unfortunately, biologists, about 100 years later, he did this in 1860, I'm sorry, I said it wrong. He made this discovery in the 1680s, like over 300 years ago. And 100 years later, biologists thought he was wrong. He wasn't wrong, but they thought he was wrong, and they thought that these glochidia were free-living parasites all on their own. They had nothing to do with mussels. They got that wrong, but they named those parasites Glochidia parasiticum, and that name Glochidia has since stuck with the larvae of the freshwater mussels. So these little Glochidia look very much like a Pac-Man. If you look up here in the upper left, you'll see an individual Glochidia, and you can see how it's got a little muscle that articulates the two halves, and these are electron micrographs, so you don't see the thin thread of a sensory hair that comes out of these, that when it touches a fish, this little Pac-Man snaps shut, either on the fins or maybe on the gills of a fish. And as it does, you see here, they've attached to this fin or onto the gill surface of the mussel. Now, once the glochidia is attached, the gill tissue forms a cyst around the glochidia and sort of tries to wall it off, but that's not enough to keep the glochidia from getting nutrition from the gills, and it doesn't grow physically in size very much. What it really does is it grows all the body parts and transforms from this larval stage into a juvenile, which has all the body parts that an adult animal has. Now, on this bottom row, we see a little fawn's foot. This is as large as the adult fawn's foot mussel from the Cahaba or anywhere else get. And this is a fragile paper shell. You can say this one is so fragile that I broke off a part of it there, unfortunately. And here is a blue-fur, another freshwater mussel from the Cahaba that uses this approach of just creating enormous number of glochidia, which sort of randomly and accidentally bump into a fish and attach to that fish and parasitize it. However, this large mussel up in the corner is called the washboard. And the washboard has a little modification of that strategy, which helps it find fish hosts. And that is that the individual glochidia are kind of strung together on an adhesive strand or a sticky hair of material that they then release into the water column. And these sticky strands then are out floating through the water column. And it's a lot more likely that a fish might run through that mesh of sticky strands. And when it does, this whole thing wraps around the fish and allows the glochidia that are attached to the strands to then snap onto and attach to the fish. So that's a little modification that helped increase the odds of um, a glochidia actually finding a fish to parasitize. That way it doesn't have to create quite the huge number of glochidia that those other species have to create. Now here we have another variation on the theme of creating glochidia that attach to a fish, and that is, this is a southern pigtoe, and this is a southern clubshell. This one is still found in the Cahaba. This one is no longer found in the Cahaba, but it is in a few other places around Alabama. What these animals do is take their glochidia and make what they call a conglutinate, which is another word for it is an ovisac. So it's made in the gills and it is full of glochidia, most of which are fertile. There are a bunch of sterile glochidia in here too that help make up the body that shapes like this. And this is pretty much a worm-shaped thing that the mother expels into the water column, and that little conglutinate or ovisac floats away, and it's something that a fish might see and say, oh, that's something to eat. It goes chopping on it and then gets a mouthful of parasitic glochidia. And these are little different, slightly different colored for the Pleuravema decisum, the southern clubshell. So here you see a close-up of one of those conglutinates, and you see there's a mature fertile glochidia, but these other, the great majority of these are only glochidia that help form the body, but it helps provide an opportunity for the fertile ones to attach. Okay, so here, that's a little modification, a little trick, a little change of the different mussels used to try to get these floating conglutinates out there as a lure. Now, here's a fine line pocketbook, and it also makes a conglutinate, but instead of floating, this conglutinate, which looks very much like a new fish egg. I don't know if you've ever seen a fish egg, but those two dark little eye spots in this spherical shape make it look very much like a fish egg. And well, predatory fish like darters and sculpins, that they find nothing better than to find a fish egg to eat. And so these sink to the bottom where the darters and the sculpins are, rather than floating away, they sink to the bottom. And when a darter bites it, it breaks open and you see thousands of the glochidia that can then get trapped on the and parasitize the gills of the darters and the sculpins. And on the right, we have an odd little mussel called the three-horned wartyback. And they call it a wartyback because the shell, it's hard to see in this profile, but if you look edge on, you see kind of a knob there and a knob there, and there are three very big prominent knobs or warts, so that give it this mussel its name, the three-horned wartyback. And rather than making a conglutinate that either floats or sinks, the three-horned wartyback makes this little white, stubby, odd-looking thing that it hangs onto it, basically. But this, for some reason, is very attractive to freshwater drum. And I don't know why they like this so much, but clearly what happens is that the three-horned wartyback makes this little white, knobby, stubby thing, and the drums come along and find it and eat it. It must be very efficient because the wartybacks spend far less energy and material making these than the other mussels we've been talking about. They make far fewer of these, so it's energetically less expensive to make this. And it apparently is very effective because almost all the freshwater drum you can find are parasitized by wartyback glochidia. Here we have a different conglutinate made by a different kidney shell species. And again, the term ovisac is instructive because you see this ovisac looks like maybe a larval fish or maybe a chironomid midge larvae. But an odd thing about this one is that the tail end here you see is sticky underwater, and that tail end will stick to a rock, so it doesn't just wash away. It finds the surface of a rock and is stuck there, and it's very likely that a darter will come along and find that and grab it and eat it. Okay, here's that ovisac that's been ruptured, and you see all the conglutinates that are coming out, and you see the little Pac-Man form. They're just ready to try to clamp down on the gills or maybe the fins of the freshwater mussel. Now, so that's some different ways that a mussel uses to trick a fish into transporting its babies around. So far we've seen that some mussels release enormous numbers of free-floating larvae, Glaucidia, that sort of randomly bump into a fish host. And then we've seen Glaucidia that are attached to long, sticky threads that will entangle and ensnare a potential fish host. Or we've seen mussels that release a floating bundle of Glaucidia or a conglutinate that mimics a worm. And then we've seen conglutinates that mimic fish eggs or other odd little things that might be on the bottom of the stream that darters and sculpins might grab. And then that the fifth one actually that we've seen are the three-horned wartybacks taking advantage of a freshwater drum's odd proclivity to eat funny little white stubby things. Now we've got, I want to talk about a completely different approach that different mussels have adopted, and that is the use of a mantle flap lure. So here we see the shell of a freshwater mussel, and here's that in-current siphon that I mentioned before, where the gills create a little current that draw water in the in-current over the gills. It filters out the food particles and then is expelled through the ex-current siphon. And that's the actual mantle itself. It helps build the layers and layers of a shell, for one thing, it functions as. But you see this right here, and you see something that looks very much like a minnow. It almost looks like it has a mouth. It's got an eye spot, dorsal fin, a tail fin. There's even sort of a dark little lateral line that mimics the lateral line of a minnow. Besides just sort of looking like this, the mussel, the female, will take this and wiggle it so that it acts like a mussel, acts like a minnow, and is even more appealing to a predator. So when a fish like a bass that might take a little minnow like that, sees it, it runs down there and grabs a hold of it. Well, when it does, it often ruptures the marsupium, where the female is raising up the Glaucidia. So here you see a lure or a lampsless, and in the center there, you see a marsupium, a specialized part of the gill that's been kind of turned over to growing up the Glaucidia. So when a bass reaches down there and bites into this, it ruptures the marsupium, and lots of the Glaucidia are released, and the fish gets a mouthful of that. So here's a different mantle flap lure that quite obviously, I'd say, mimics a crayfish. And you see the eyes, you see the legs, you see the tail flipping like a crayfish does. And beyond simply looking like a crayfish, the female has her foot planted in the stream bed, and she swings her whole shell so that the crayfish looks like he's swimming backwards like crayfish do. Now, here on the left, we have a mantle flap lure of a fine line pocket hook. And this, I would argue, is a pretty good mimic of this little guy. Well, not little, this is probably at least 3 inches or more. This is called a helgramite, or a Dobson fly larva. And it's obviously a predatory insect larvae that feeds on other insects. It's about the best fish bait you can find. If you can get fishermen often get a window screen and a couple of poles and go kick over rocks so they can collect a few helgramite larvae to bait with, because it's almost certainly going to get you a bite from a bass or something like that. Now, in addition to this mantle flap lure, the fine line has an additional strategy, and that is that it can also make a super conglutinate. So we talked about conglutinates before, when a mother can create 50 to 100 little bundles of her Glaucidia in little bundles and little packages and release. In a superconglutient, all of the Glaucidia are bundled into a structure that looks like a minnow. And here we have an example of that. So here's the muscle. You can't see the clear tube, but that is the superconglutient of the muscle. There you can see the tube, and it looks pretty much like a minnow swimming around in the water. It's really a pretty effective lure, and it's something that if you look on the web, you can find videos of bass grabbing hold of one of these. Now, so far what we've described are, I guess you'd say, passive lures. They attract the fish, and if the fish is sufficiently deceived, it might take a bite out of a conglutient or a superconglutient or a mantle flap lure. But here we have something different going on. This fish is called the Mobile log perch, and it has kind of a long snout, and it's kind of, as fish snouts go, it's kind of hard and tough. And the reason is that the log perch has this habit of nudging rocks and flipping over rocks, because insect larvae, they don't hang out on the top of rocks. They'd just be fish bait if they were. They hide underneath the rocks and down in the crevices and gaps and spaces between the rocks. So the Mobile log perch is successful because he'll nudge the rock and maybe turn it over, and the fish larvae will float away, and then they grab it. However, we also have the comb shell species of mussels that take advantage of that fact. And what they do is, if a log perch comes along and tries to turn over a comb shell, the comb shell will grab him by the nose, and then he has a set of teeth along the outside edge of the shell that can help hold it. And then the mussel starts pumping larvae into the mouth of the Mobile log perch, and it gives a pretty heavy infestation on the gills. So here we have a comb shell, and you kind of see some teeth right along there that are going to be useful here in a second. So a log perch, he sees something interesting, he starts to turn it over, and snap. The comb shells grab the hold, and this poor log perch doesn't know what to do. He's he's flailing around at this point, and he's trapped. So what's happening now is that the comb shell is starting to pump larvae into the mouth of the fish and out the gills, and gets a pretty heavy infestation of, whoops, let me back up, sorry, of the poor little fish. Here's a different kind of mussel in that same group. This is called a riffle shell. And I'm not sure what it is that's appealing about this little white feathery thing, but apparently it's something that is attractive to fish, and maybe at least they'll come and investigate. Here comes a little darter. He's looking around, and oops, there's the riffle shell grabbing a hold. And not only does this shell and this shell clamp down, you also see this sympallium membrane that also helps form the seal around the poor little darter there. And at this point, the poor little darter is struggling. It can't even breathe very well, but it would be a really bad idea for the mussel to kill the darter, because then it wouldn't move its little babies around. So it begins to relax its hold, and then you see here the Glaucidia, little bits of Glaucidia that the darter is beginning to suck in or over its gills, and getting an infestation of these little parasites on its gills. So now the fish is beginning to percolate well, and he's starting to recover. And actually the mussel is going to try to kind of push him out and blow him out. So now the fish, with its new supply of parasites on its gills, is ready to swim away. And after 3 or 4 weeks, the little Glaucidia will rupture the gill and fall out, and hopefully it falls into the right kind of habitat. Now, in order to enhance the chances that this will work out, the mussel is trying to attract a kind of fish that hangs out in the right kind of habitat. All these mussels have their own unique habitat, and they want to enhance the chances that a larvae will fall, the juvenile, transformed juvenile will fall out in the right habitat by attracting a fish that hangs out in that habitat. So that's part of the strategy of why they make the particular lure that they do. Now, finding a fish host is not the only problem that freshwater mussels have to face. Sometimes we've seen in our streams with heavy sediment loads that deposition of sediment can smother habitat. And maybe even more importantly, since these animals are filter feeders, they can't just jump up and run away when a water quality problem comes along. They've got to somehow endure that. So anytime there's a water quality issue, they can only just breathe minimally and not feed, and they're just very sensitive to water quality conditions. And that's why we've seen such a high proportion of these freshwater mussel species that have not survived the water quality that we're creating these days. And the last problem that I'll mention is a drought. You see here a southern pocketbook that is experiencing a sort of a low water problem. Here's the in-current siphon and the ex-current siphon. This poor little guy can't even breathe at this point. The water level's gotten so low. And it's not just that they can't breathe, but I'll come back to that. But so some mussels have adapted to the fact that water levels in rivers change. And here you see a groove where a mussel has managed to crawl down slope and made it back to water. So he's back in this shallow water here, but at least he's not up here on the bank anymore. But sometimes the slope down to deeper water is not very obvious, and they just kind of wander around not knowing where exactly to go. And if they're lucky, they make it to deeper water, and if they're not lucky, they don't. Here's one that started up here on the bank, and he's kind of meandered around. I thought that one was kind of interesting. Now, it's not just that they have to stay in the water, but they have to get in deeper water, because any mussel that makes it out here is still in shallow water, and raccoons and muskrats and other things can find them. So if they don't make it to deep water, they become prey to a lot of different animals that can eat them. Well, mussels aren't the only interesting mollusks that we have in our rivers. Snails are another group of critters, and here again is a map of the number of freshwater snails by state, and you see that Alabama has far more snails, and this may be 300 rather than this when this map was made, species of snails in Alabama. In the Cahaba, we have 31 different kinds of snails. Here's a federally endangered snail called the cylindrical Lyoplax. Here's an endangered species called a flat pebble snail. And this one in the lower right is called the Cahaba pebble snail. And it's interesting because it was described in the 60s, and by the 70s, they couldn't find it again, and they declared it extinct. And about 2005, Dr. Stephanie Clark rediscovered this Cahaba pebble snail in the Cahaba River National Wildlife Refuge. And so it wasn't as extinct as we thought it was, which is nice. Now, this thing is really quite, maybe the reason that it wasn't found, it's quite small. It's hard to recognize, hard to find, and she was able to find it and describe where it was hanging out, and now other people are able to find it. So the Cahaba pebble snail is actually recovered from extinction, if you will. Another snail species that we thought was extinct is the oblong rock snail. And this is a snail that had not been seen for 70 years when graduate student Nathan Whalen and Dr. Johnson at the Alabama Aquatic Biodiversity Center decided they'd go look back again at the locations where it had been found in the past, and these little squares mark those locations. And sure enough, they were able to relocate this oblong rock snail again. So it's the second snail species that's been found to not be extinct. in the Cahaba. This is a summary of reintroductions of mollusks that were done by the Alabama Aquatic Biodiversity Center. This is part of the Alabama Department of Natural Resources and the Fisheries Division. And what they do is go find the very last living examples of a number of mussels and snails, and they're able to rear them through their life cycle and get them to parasitize a fish, uh, get the transformed larvae off the fish, and then keep them alive and growing until they're big enough to be carried out and reintroduced in places around the state. So these are all the relocation sites that were used in 2012. The Cahaba is one of the favorite places to reintroduce things because the water quality is good enough, particularly in the south half of the river, to allow all the work that goes into rearing these things out. You don't want to put it into a dirty river. Crayfish are another example of an aquatic group that is just crazy species rich in Alabama. We have something like 98 different crayfishes species in the Cahaba. This says 19. I think the actual number is 17 crayfish species. Well, native crayfish species. What we're not including in that number 17 is this is called the devil crayfish. It's an invasive species, and it actually will displace the other native species. So it's not necessarily a good thing to find one of those guys. 17 turtle species. No, it's more than that. It's there's about 20 some turtle species in Alabama. This is a map turtle and a snapping turtle and a stink pot or a musk turtle. And of course, we have the good old box turtles. So Alabama is really rich in turtle species. And for the other aquatic groups, in the insects, the dragonflies and mayflies and damselflies, this thing in the lower left is actually a freshwater bryozoan. And they're microscopic, but they form this gelatinous matrix that they live in. So this is populated by thousands of microscopic filter feeding animals, and they exude this almost like a tough jello layer that they live in, and that's usually wrapped around some twigs and sticks and so forth. So they stay put. You'll find these in slower moving water situations, and that's an odd thing to find. Some of them are the size of a basketball or so. And of course, we've got river otters and muskrats and other kinds of aquatic things. Well, that's our little tour of aquatic biodiversity. We didn't say anything yet about the weird, wonderful, newly discovered plants on the Big County glades, or about the Cahaba lilies, and perhaps in a future episode we will cover that topic. But I hope you've enjoyed learning a little bit about the weird and wonderful things, the beautiful things that live in the Cahaba River, and we hope we'll see you again next time. Thanks.